So good morning students. In the last class we were discussing about the law of dominance. Okay, you had studied the law of dominance. The next second law, what it is means that it is the law of segregation. Let's see about the law of segregation. Law of segregation. In this law, what this law states is it states that whenever a pair of factors, it states that whenever a pair of factors, pair of factor for a character is brought together for a character is brought together in a hybrid they separate or segregate during formation of gametes. Okay, so here what this law of segregation states means whenever a pair of factor, for example, here factor means that is the genes. For example, for a particular tall, this is a pair of factor. What is tall? This is one of the height. What is height now? Height is the character. What is height? It is one of the character. What is that pair or factor or genes which is responsible for this height means that it is the capital D and capital D. Okay, so that particular they will be present in what pairs, but during gamete formation they will separate. They will get separated. During monohybrid cross, what you had studied, the parents will be capital D and capital D. Okay, which is crossed with the small t and small t. Okay, so when they are going to see, when offspring is going to form, when the gamete are going to fuse. So at that time, during gamete formation, they will get separated from each other. These are the gametes. Okay, so see here. Whenever a pair of factor for a character is brought together in a hybrid, okay, they are getting together in a hybrid like this. This is the hybrid of F1 generation. For next self cross, they get separated. How means this will get separated in this way. Either they will get separate or segregate, means they are going to divide when during the formation of gametes. So this is the law of segregation. Okay, the next after law of segregation, next comes the next topic, that is the incomplete dominance. next topic, that is the incomplete dominance. Incomplete dominance, which is also known as the partial dominance. Partial dominance are blended inheritance. So this question will be asked for 5 marks. Incomplete dominance, which is also known as the partial dominance. This question will be asked for 5 marks. Okay, so here, what do you mean by, what is the definition of incomplete dominance? Let's see here. It is a process where the dominant gene is a process where the dominant gene is incompletely dominant. Gene is incompletely dominant over the recessive gene. Incompletely over the recessive gene. And produces a phenotype which is intermediate and produces a phenotype which is intermediate to the parental type, which is intermediate to the parental type. This is the definition of what? Incomplete dominance. Here you see, it is a process where the dominant gene, which gene? Dominant gene is incompletely dominant, not completely. It is incompletely dominant over the recessive genes. Okay, see here, from first we are taking as an example of height. One is tall, one is dark. Okay, here the tall, capital T is dominant, tall is the dominant, and small t is the recessive. Okay, the tall gene is incompletely dominant over the dark one. Okay, in the recessive, and they produces a phenotype. Means they both will produces a phenotype which is intermediate to the parental type. Intermediate means in between, not tall, not short, medium sized. Okay, here you see tall when it is crossed with dark. Okay, tall is the dominant, dark is the recessive. We know that. If we cross them, we will get medium sized height or medium sized plant. What is this? This is the in between the tall and dark. Why an intermediate type offspring is going to produce here means because this tall is incompletely dominant, not completely. If it is completely dominant, here whatever the offspring will be there, that will be the tall one. Okay, but here it is a medium sized plant. So by taking one of the example, here I will explain the incomplete dominance or partial dominance. Means incompletely dominant over the recessive gene. See here, when experiments on peas were repeated using other traits in other plants, okay, during incomplete dominance, accept the height. Except till now in monohybrid cross, in test cross, we have taken, in monohybrid cross we have taken the height as a character. In test cross we have taken the color of the flower. Now next we will take again the color of the flower, okay, but not in the pea plant, but in some other plant. Okay, so it was found that sometimes the F1 or the F1 generation and phenotype, they did not resemble either of their parents. Okay, means they are not similar to their parents, they are different, means because they are going to form an intermediate type which is not similar to their parents. Okay, so they had taken they had taken the example, the inheritance of flower color in the inheritance of flower color in one of the plant species name is there, that it is the snapdragon. In snapdragon or antherinum species. Snapdragon or antherinum species. This is the name of that particular flower. What it is? Snapdragon or antherinum species. It is a good example to understand the incomplete dominance. Okay, and which is also known as the, one more plant is there, that is Mirabilis jalapa. Mirabilis jalapa, which is also known as the 4 o'clock plant or 4 o'clock plant. This is also one of the example to understand a correct explanation of what? Of the incomplete dominance. 2 examples are there, snapdragon or antherinum species. One more example, Mirabilis jalapa or 4 o'clock plant. Snapdragon, 4 o'clock plant are the common names. Antherinum species and Mirabilis jalapa are the scientific names. Okay, so let's see about this plant. By taking an snapdragon or antherinum species, we will cross here and we will understand the incomplete dominance. Okay, so in a cross here, they had taken first parents. I will rub this statement. Here, first parents. We have to take, in which plant we are taking here? That it is in the snapdragon or antherinum species. This is very important point. Antherinum species. Don't forget to mention the example in answer. Okay. Sometimes they will ask to explain the incomplete dominance in snapdragon plant. Okay. So like that questions will be asked. So here you see first we will mention the parents. What parents they are going to take here means they are going to take the color of the flower. They are going to study the incomplete dominance in by taking what character, that is the coloring of flower in snapdragon or anthurium species. First, which color? That it is red color. Red colored flower are crossed with, here I will write, red color flower crossed with white colored flowers. Okay, that means the plant which is having the red color flower are crossed with the white color flowers. Okay, so here you see it can be represented with capital R and capital R, and this can be represented with small r and small r. Okay, parents, red colored flower which is represented by capital R and capital, what it will become? Capital R, small r. But here capital R is dominant, small r is recessive. Okay, but here the capital R is not completely dominant. If it is you would have said the color of the flower is red color, but it's not red color. Here the genes are going to mix up. Okay, that's why here the red color mixes with the white color and produces a pink colored flower. Okay, what they will produce? They will produce the pink colored flower. And what it is? This is the F1 generation. What is the offspring? After crossing red color with the white color flower, it comes the pink color flowers. Okay, then again we have to self cross. Here self cross has to take place. Here I will show you that one. Self cross. See here, whatever the F1 generation offsprings we got, that is capital R, small r, it has to cross with the same plant. Capital R, small r. So these we can say it as the self cross. Okay. The next here, what will be the gametes now of F1 generation? This will be the capital R, small r into capital R, small r. By using the unit square checkerboard, we can get the F2 generation. Here you see capital R, small r, capital R, small r. This is the male and this is the female. Okay. Now next you see here capital R and capital R, capital R, capital R. Okay. Next capital R into small r. It will become capital R, small r. Okay. The next small r into capital R, that is capital R, small r. Always the capital letter should be written first. Then next you have to write the small letter because the capital R is the dominant one. Small r into small r, small r, small r. The color here. caterpillar, both are dominant. What it will be? It will be the red color flower. Okay. The next, caterpillar is smaller, it will become pink color, not red color. Because this R is incompletely dominant over the smaller. Okay, incompletely dominant means they are going to make some new offspring, which is not identical, which is not similar to their parents. Okay, so it will become pink color. Here also pink color, and here they both are recessive. Recessive means they both are not going to express their characters. So what it is? Smaller, that it is white color. Now next the question comes off about a ratio. Phenotypic ratio. Here I will write the phenotypic ratio. Otherwise here I will write phenotypic ratio. That it is 1. 1 is red. Okay. Pink is how many? 1, 2. Okay. 1 is to 2. Then white is 1. Here I will write 1 is red, 2 pink, and 1 again white. This is the phenotypic ratio. 1 is to 2 is to 1. Otherwise I will write here. Can't see there. Phenotypic ratio. One more ratio, that is genotypic ratio. Okay, here phenotypic ratio means 1 is to 2 is to 1. Okay, 1 is to 2 is to 1. 1 is red color, 2 is pink color, and 1 more is white color. Phenotypic, by seeing the external character, that is the color of the flower, pink, red and white. One more thing, that is about the genotypic ratio, by seeing the genes. Here caterpillar, how many are there? This type of genes, only 1. Okay, 1. Pink, caterpillar smaller, how many are there? Only 2. And smaller, only 1. So this is the phenotype and genotype ratio of the incomplete dominance. 1 is to 2 is to 1. Genotype is also 1 is to 2 is to 1. Okay. So this is about what? Here red color, pink color, white color. See, in monohybrid cross, one gene is going to inherit it from the parents to the next generation. In that case, what happens means, if one gene is inheriting, that one gene will be dominant over the recessive one. If like that are there, dominant over the recessive, they will produce the same characters which is present in the parents, tall or dark. But here a new character, a new flower is going to form. Which colored flower? That is the pink colored flower. See, what is the meaning of incomplete dominance or partial dominance? Means it is a process where the dominant gene is incompletely dominant over the recessive gene. Okay, dominant gene is incompletely dominant over the recessive gene. That means red colored flower is incompletely dominant over the white colored flower. Okay, and they are going to produce the intermediate type of offspring. Intermediate means if you mix the red color with, by taking the paint, generally I am saying, if you mix red color with the white color, you will get the pink color. Okay, like that only, their blending, mixing up is going to take place. Mixing up of what? Of genes, which is going to inherit it to the next generation. Okay, so here red color is incompletely dominant over the white color. After that, which will produce a phenotype which is intermediate to the parental type. Here the parents are red and white. Okay. Here during the gamete formation, they will get separated. What? That is the genes. In the F1 generation, whatever the offspring is produced, that is intermediate. Intermediate means in between the parents. In between the red and white color, which color will come? That it is the pink colored flower. Okay, then again, what we are going to do? This is the offspring of F1 generation. We will take that offspring, then again we have to cross with the same plant. That means pink colored plant or pink colored flower. If we made them, here you see, that process we can call it as the self-cross or self-pollination. What we can call? We can call it as the self-cross or self-pollination, where with the same parent we have to cross. Okay. So here, what will be the gametes for them? Capital R, smaller, capital R, smaller. Don't forget these steps. These steps are very important. If you write these steps or if you represent this correctly, then you will get full 5 marks without any mistake. Okay. If asked to explain incomplete dominance by taking snapdragon or 4 o'clock plant or Erythrinium species or what we call Mirabilis jalapa, if you explain this much, it's enough. Okay. See here, these are the gametes. One parent gametes we have to take one side, another parent gametes we have to take another side. Then we have to cross. Confusion during cross. Always you have to write the dominant character first, then the recessive character. Okay, capital R into capital R, capital R, capital R. That will become red colored flower. Okay, capital R into small r, that will become pink colored flower because this is an intermediate type of phenotype. Okay, of what? Of the parents. Small r into capital R, small r, that is also pink color. Small r into small r, that is white color. So what will be the phenotypic ratio? 1:1 red color to pink color, 1 white color. This is the phenotypic ratio. Again the genotypic ratio, 1 red color to pink color and 1 white color. Then again it is the 1:2:1. Okay, so this is about what? About the incomplete dominance. Okay, the next here, what is the explanation behind the concept of dominance? How the genes are going to incompletely dominant over the other, that we have to see. Okay, we will see that one. Please note it down, this representation, which is very important. Now next you see here, explanation of the concept of dominance, that we have to see. Explanation of the concept of dominance. Here you listen here carefully. Why that gene is incompletely dominant over the recessive gene? See, every gene expresses itself by producing a particular protein or enzyme. Here I will write, every gene expresses itself. In what way they will express? They will express in a form of character. For height, there will be one gene. For color of the flower, there will be one gene. For color, shape of the seed, there will be one gene. Like that. How they are going to express? That genes will have a protein. Okay, these genes which are expressing itself, that gene by itself, they are going to produce a particular protein. By that protein only, that particular plant is going to get the character. They are going to express their character. See, every gene expresses itself by producing a particular character. It's a protein. Protein, or it may be enzyme. Both are same. Protein or enzyme. See, this is the gene. Gene for what? Gene for color of flower. Color of flower is what? This is the character. What it is? This is the character. How this character you are seeing in a particular plant? That character is coming through the gene. What that gene will have? That gene is going to produce a protein. That protein is going to form a particular character. Okay. So that particular protein or enzyme, how they are going to act in the dominance. So see here. That particular protein or enzyme acts on a particular substrate. Okay, this enzyme will act on a particular substrate. Then the reaction is going to take place with enzyme and substrate. Particular substrate. This substrate can be represented as the capital S. This enzyme is going to bind with the substrate. Okay, from where this enzyme or protein is going to get? That is from a gene. Okay, so which brings one expression for a particular trait. When this enzyme binds with the substrate, then it brings a trait. It brings a particular trait or character. This substrate will get bind with the enzyme, then they both will express what? A particular trait or character. They are the similar thing, such as the color, height, shape of the seed. By taking only pea plant or mirabelle jalafa, we can say these all are the characters. But in human beings also, whatever the character, such as the shape, height, color of the hair, type of the hair, all the characters are due to the protein or enzyme, which will bind with the substrate, and they both will express, and then they are going to form a particular trait or character. Okay, so this is about in this way. And one more thing, during this binding and all, a normal allele will produce a normal enzyme. Normal allele means what? Here I am telling every gene expresses itself. Genes will be present in pairs. These are the two genes which are present in pairs, which we can call it as the allele. If this allele is normal, or normal allele is there, this normal allele will produce a normal enzyme. What does it produce? If allele is normal, it produces a normal enzyme. A non-functional enzyme, or it may not produce a non-functional allele is there, means they will not produce any enzyme, then there will be that gene is not responsible for any trait. Okay, so for example, one more thing, if change a non-functional enzyme or no enzyme, then it will be not be able to produce a phenotypic expression. See, If this allele is not going to produce any enzyme, then there will be no particular trait or character. Okay, so during which the phenotypic means that is the external characterism. So the phenotypic depends, what we call it, phenotypic expression, it is mainly depends only on the functioning of the genes or alleles. Okay, so the next thing, hence the whatever the here the alleles of two types. What are that? Alleles are of two types. One is modified allele and another is unmodified allele. Unmodified allele will not produce the normal enzyme. If there is no production or there is no expression of a normal allele, then there will be no particular trait or character. Okay, so in this way, see, hence the unmodified allele responsible for the original expression of original phenotype, and this unmodified allele we can call it as the dominant allele. This we can call it as the dominant allele, and modified allele we can call it as the recessive allele. Because the unmodified allele will not produce any enzyme for a particular trait. So here both the alleles, the genes are going to mixed up, by which they are going to form the by which they are incompletely dominant over the recessive gene. So this is about what? About the incomplete dominance. Next topic is codominance. What it is? That it is the codominance. Here they had taken one example, that it is the blood group of human beings. Okay, as basically you know, there are four types of blood groups: A, B, AB and O. Okay, certain antigens, antibodies are responsible for these four types of blood groups. But what is the thing means, there are some genes in the cells, okay, which is responsible for these blood groups. What is there? There are some genes or alleles which are responsible for these blood groups. Okay, so here codominance means where both alleles in a heterozygote condition expresses themselves equally. Heterozygote means where the two genes in a particular allele, they both are differing from each other. They both are differing and they both are expressing on each other. Okay, they both will be dominant on each other. That means the codominance. What it is? That it is the codominance, where both the alleles in a heterozygote condition, they both will express themselves equally. Here in the incomplete dominance, one allele or one gene is incompletely dominant over the recessive. But here in the codominance, both the alleles are dominant over each other, or both the alleles are expressing on each other. Okay, so this is about what? About the incomplete dominance. In the next class, we will continue with the codominance. Okay, by taking an example of ABO blood group in the human as an example. Okay, thank you.